All right, so first thing we're going to do is talk a little bit about animals, do a little bit of review into that. Here's our phylogenetic tree. We know it's a tree that shows what two things? How things are related through evolution and genetics, phylogenics. So far we've talked about eubacteria, the first modern living thing to come off the branch. We talked about archaebacteria, we talked about protists. Protists were supposedly the first what? First what? Just look at the things. What were they the first of? Eukaryotes. Single-celled eukaryotes, like algae and protozoans, like amoebas and parameciums. And they are supposed to be the ancestors of all the modern-day prokaryotes, things like fungi, animals, and plants. Now, in terms of animals, this is the characteristic of an animal cell. Eukaryotic, multicellular, heterotrophic. All three of those first things also apply to what? Those first three terms all apply also to what other kingdom? Again? Nope. Third one does it. That's what we're talking about. What other kingdom do all three of those first three things apply to? Fungi. The differences start right here, where animal cells don't have cell walls. Fungi do. Their cell walls are made out of, usually, chitin. Animal cells, or at least the organisms, are usually motile. They can move on their own. Fungi usually can't. Our cells as animals are almost always diploid, meaning we have two sets of what? Two sets of chromosomes. Fungi usually aren't. They're usually haploid. They usually only have one set of each chromosome. So that's just sort of a review to compare and contrast animals and fungi. Now, what we're going to try to focus on today is this embryonic development stuff. Let's keep this fairly simple. The first thing that happens in an animal, before I do that, let me talk about this word right here, metazoan. Anybody know what that is? Metazoan? It's an animal with true tissues. Almost all animals fall into that category. I can only think of one exception to the rule, and that's a sponge. A sponge is just a huge group of cells with pretty much very little order to them, not any real tissues. Metazoans are pretty much every other animal except for a sponge. But we're going to talk about how their embryos develop. And in the first most important step, maybe, is this step right here, fertilization. We start off with a haploid sperm and a haploid egg. Haploid plus haploid eventually will give you what? Diploid. And that first cell, that first diploid cell that you come from is called the zygote. That word's an important word to know. The first cell of a new baby, sperm plus egg, gives you a diploid zygote. As soon as that happens, the egg has been storing up things, proteins, RNA, ready for this event to happen. And when it does, once fertilization happens, that triggers that cell to divide really, really quickly. It divides so quick that the cells don't have any time to wait. Don't have any time to grow. So you start with one cell, and then in just a little while you have a bunch of cells, but it hadn't gotten any wet. It hadn't gotten any bigger, it's just divided itself up into different cells. What process of cell division are we talking about here? Mitosis. Meiosis already happened, right? Meiosis creates what? What's the job of meiosis? To create what? Well, yeah, but what kind of cells? Sex cells, sperm and egg. That's been done a long time ago already by this point, right? Mitosis is what's going to happen from here on out. Another thing that happens during this part of the embryonic development stage is the cells start to orient themselves, align themselves on three what are called the embryonic axes, anterior and posterior. What's what the anterior means? Anterior. If you're a four-legged dog, where's your anterior side? The front of you. So it'd be like the head of the dog. Posterior, therefore, would be what? The back. Dorsal ventral. If you're a four-legged animal, where's dorsal? Yeah, the top, where your backbone would be, right? Think of like a dorsal fin in a shark. That's easy to remember. Ventral, therefore, must be what? By the belly side, the bottom side. And then left and right. So in fertilization, haploid sperm plus haploid egg gives you this diploid zygote. That cell has the egg cell has been preparing for this. So once it happens, it starts to divide very rapidly. It starts to orient the cells on three main axes. All right, then we get cleavage. Cleave simply means to like a butcher uses a cleaver. What's a cleaver? Cut, split, break, tear. So again, it continues to divide into smaller and smaller cells with no growth of the embryo. It's splitting so fast that the cells don't have any time to grow. So we're getting more cells, but no more size. The first thing that happens here is the cells eventually form a solid ball of cells. And that solid ball of cells, term you need to know, is called the morula. The solid ball of cells is called the morula. Some people say morula. After a little time, these cells secrete this thick liquid. And this liquid basically forces a cavity in the middle of the cells. So it goes from being a solid ball of cells to all of a sudden that thick liquid has hollowed out a what? A cavity, almost like a tube.
Like hydrolytic enzymes or something?
No, it's just pushing them out of the way. So you end up next with this cell that's like a hollow ball of cells. And that hollow ball of cells is called the blastula. So you start off with a solid ball of cells called the morula. Eventually this thick liquid hollows it out, and you've got this cell called the blastula. What's going to happen next is the cells are going to start to move around, and they're going to start to organize themselves. Mostly, we're talking about the cells on the inside of this blastula that do that. What do you think the cells on the outside of the blastula become? Cells on the outside of the ball, what do you think they make? They make stuff like the placenta, the umbilical cord. Those kinds of things are made by the cells on the outside. The cells on the inside of the ball actually make what? Outside make the placenta, inside cells must therefore make what? What we're trying to make here, about a baby. Cells on the inside make the baby. But these cells on the inside organize themselves into three layers. Sometimes you'll hear them called tissue layers. I'm going to call them the germ layers. The cells on the inside of this ball of cells organize themselves into three layers called the germ layers. These cells are your stem cells. That's an important word to know. What are stem cells? Somebody in here knows what stem cells are. What are they? Yeah, they're cells that haven't specialized yet. They haven't decided what they want to be when they grow up. So they have the ability to become what? Anything. These are the cells that will make you, the cells that will become you, the cells that will make certain parts of you. Before I go any deeper in that, there's a lot of interest in science today in stem cells. What are they trying to figure out how to do with them outside of the body? Trying to figure out how to use them to fix certain problems. Well, for example, Christopher Reeve. You guys know who Christopher Reeve was? It's the guy who was Superman in the most famous set of Superman movies. The actual actor. Yes, like there you go. What happened to Christopher Reeve, the actor? He fell off a horse and became paralyzed. And he became a big advocate for stem cell research because one of the things they're trying to do with stem cells is, your nerves, once you're grown, they don't do much growing. They don't divide very much. So if you sever your spine, is it going to fix itself? Probably not. So what a lot of scientists are researching how to do is how to use stem cells to regrow and repair nervous system damage, brain damage, spinal cord damage. Well, they're trying to figure out exactly how to do that, how to make them do what they want them to do. Because they have the ability to do what? Anything. So scientists have to figure out how to turn on the right switches to make them do what they need them to do. Fix the spine. They looked into curing Parkinson's disease, which is a problem with your brain, causing you to twitch and stuff, right? Trying to use stem cells to fix that. Leukemia, which is a problem with a part of your body. Your bone marrow. And actually there's a lot of people today who, when the baby is born, what part of the baby do they keep? In case there's problems later. Not. They keep the blood in the umbilical cord. They store it. Because what's in that umbilical cord blood? Lots of stem cells. Keep those, and it's already been proven that those stem cells can be used to cure people with leukemia. Because you can put those into the bone marrow and they will start to create normal red blood cells, white blood cells, that kind of thing. If it's a match, if they match up correctly. So stem cells are really important. These are your first stem cells, the stem cells that made you. But the first thing they do here in this stage called gastrulation is they organize themselves into 3 layers. 3 layers of stem cells. The innermost cell in that ball is called the endoderm. The middle layer in that ball is called the mesoderm or mesoderm. And then the outermost layer of those stem cells is called the ectoderm. Once these cells organize themselves into 3 layers, the ball now is called the gastrula. So we went from a morula, which is a solid ball of just a few cells, to a blastula, which is the hollow ball of cells that will become the baby eventually, all the way up to the gastrula, which is the ball of cells that's organized itself into 3 layers of stem cells. Three layers of stem cells called the germ layers. Now I want you to realize that these 3 germ cell layers, each one of them is going to be in charge of making certain parts of your body. And we're going to look at that in a minute. Actually, let me go a little bit further here. Before I go into organogenesis, I'm going to skip up a slide. Here's the blastula. You can see it's hollowed out. Right there, it's a hollow ball of cells. Here's a gastrula. Now, it's hard to see the layers of cells, but you've got a layer on the outside. That's the ectoderm. You've got a layer of cells on the inside, endoderm. And you're also going to have a layer here in the middle called the mesoderm. Another thing I want you to see is that this cell has also started to invaginate in, to make a tube inside itself. It's got an opening. That opening is called the blastophore. What do you think that becomes? The opening into this ball of cells. Yeah, it depends on what kind of animal you are. If you're a human, that's your anal opening. Most other animals, that's what? That's the mouth. Depends on what kind of animal you are. But in most things, this becomes the mouth. But in us vertebrates, it becomes the anus. Also in starfish, it becomes the anus. Exactly. And this tube, what does that tube become? Well, if that's your anus, what's the tube? The whole digestive tract. Sometimes that's called the primitive gut. Sometimes that tube is called the arc enterine. But that becomes your digestive system, essentially. The tube that makes up your digestive system. So again, the gastrula is a ball of cells in the embryonic development stage that has organized itself into 3 layers of stem cells called the germ cell layers. The gastrula has an opening in it and a tube. That opening is called the blastopore. And depending on what kind of animal you are, that's either going to become the mouth or the anus. And the tube, called the arc enterine, is going to become your digestive system. The last stage that we'll mention in embryonic development is organogenesis. What's genesis mean? Creation. So this is the creation of your organs. Your stem cells do this. Each of those different layers of stem cells, ecto, meso, and endoderm, those create your organs for you. Like it says here, for vertebrates, one of the earliest things that happens is development of what's called the neural tube. What's the neural tube going to be? What does it make sense we would make first? Your brain and your spinal cord, your nervous system. It says here that becomes the central axis of the embryo. We're sort of built around what? What does it make sense we might be built around? Spinal cord, right? Your brain and your spinal cord sort of built around that, the neural tube. And then after that, your major organ systems are made at about the same time. It doesn't take long. So the 4 stages of animal embryonic development. Fertilization. Egg and sperm, haploid egg, haploid sperm get together, make a diploid zygote. That diploid cell very quickly starts to cleave, it starts to split. It's splitting so fast that you're getting more cells, but really no more size. Those cells eventually organize themselves into something called a morula, a solid ball of cells. Fairly quickly, they start to secrete a thick liquid that hollows out the cell, hollows out the ball. Once it becomes a hollow ball of cells, that's called the blastula. The cells on the outside of that blastula, we said make what? What do the cells on the outside make?
Your
Placenta, umbilical cord, that kind of stuff. The cells on the inside actually make the baby. Those cells organize themselves into 3 layers of cells. 3 layers of what kind of cells? Stem cells. Stem cells that haven't differentiated yet. Meaning, they haven't decided what they want to be when they grow up. Meaning they have the ability to become what? Anything. And they organize themselves into 3 germ layers. Ectoderm on the outside, mesoderm on the middle, and endoderm on the very innermost part. Once it gets organized into 3 layers of cells, 3 layers of stem cells, the ball of cells is called the gastrula. And then lastly, we get organogenesis, where the organs are created. But I want to show you 2 other quick things of this, and then we'll go back and we'll do a little bit of reviewing for the midterm. Just in general, here's some things that these germ layers do for you. The endoderm, the innermost layer of stem cells, makes stuff on the very innermost part of your body. Kind of makes sense, right? The digestive tract, that's sort of the innermost part of you. Your lungs are very inner. Your liver, your pancreas. Those are things made by the innermost layer of stem cells. the endoderm. The mesoderm, the middle layer of stem cells, makes stuff that's sort of in the middle of your body. Things like the dermis. What's the dermis? What part of the skin?
The part under the
This is mesoderm, so what part of the skin? Mesoderm. What's meso mean? The middle layer of the skin is the dermis. The bones, which are sort of in the middle of things, right? The middle of your arm, the middle of your leg, the middle of your body. That's where your bones are at. The cartilage, the blood vessels, the kidneys, gonads are your reproductive organs, your muscles, and also the linings of most of your body's cavities. For example, what's this cavity right here called, where your lungs are at? Thoracic cavity. What's this cavity down here called? Where your stomach is at. Got this area of your body, what is it called? Abdominal cavity. Go a little bit further down, cavity down here is pelvic cavity. Cavity up here is head cavity. Let's be a little more scientific sounding. What's the name for this area? Keep going. But the cranial cavity. So cranial, thoracic, abdominal, pelvic. Right, and then lastly, the ectoderm, the layer of stem cells on the outside, those make stuff that are sort of outer parts of your body mostly, like the epidermis, which is what? What part? The outermost part of your skin, part you can see. The nervous system. A lot of your sense organs, like your ear and your eye. The tooth enamel, which is what part of your tooth? Outermost part of your tooth. The epithelium of the mouth, which would be the lining inside your mouth. Your nose. And this is saying the epithelium of the mouth, the nose, the salivary glands, and the anus. So the lining of those things, the outer lining of those pieces. All right, again, so these three layers, endoderm, mesoderm, and ectoderm, they are what kind of cells? Stem cells. Cells that haven't differentiated yet, but in this process are starting to differentiate, starting to make specific parts of your body. All these cells are related how? If this was happening in you, creating you right now, how are all those cells related? Genetically, how are they all related? Every cell in your body is related how? They are identical because they all started from what? One what? No, started from two gametes. Started from one zygote. And that one zygote created all the cells in your body, all the trillions of your cells in your body by what process? Mitosis. So genetically, they're all the same. So how come they're all different? What makes cells in your kidneys different from cells in your brain, and different from cells in your lungs, and different from cells in your toes? Yeah, but what makes them specialized? How do they look different, even though they're genetically the same? Yeah. Physically, what makes them be different, though? Well, it is because they have different proteins, because different what are working? Because different what are working? Different genes. Even though they all have the same genes, are the same genes flipped on in all the cells? No. That's what makes them differentiate. And if scientists can figure out how to manually flip on and off certain genes in stem cells, they ought to be able to make what? Anything they want to make, like replacement organs. That's where this is going. I mean, that would be pretty awesome. If you needed a kidney transplant and all of a sudden they just take a few cells from you and create a new kidney. It'd be pretty cool, right? And that's where it's going. That's going to happen probably eventually. All right, that's where we're going to stop with this for right now.
No,